Oh, it's hot here as well.
That means the mics are alive. Oh, crikey. Can't see now. Right? Good morning. Good morning, everybody. If you'd like to take your seats, we'll commence our session. So it gives me very great pleasure to welcome you on behalf of my co-chair, Dr. Lena Hjermind, to this session entitled Update on Planned Clinical Trials. As you will have seen in the previous session before coffee, there's a wide variety of ongoing clinical trials. But the good news is there are even more clinical trials in planning. In fact, in my last count, we had 15 programs, each of which have clinical trials in planning, of which we've had to choose 6. So we have a 60-minute session, 6 presentations. So unfortunately, because we're squeezing so many presentations in, we will be unable to accompany or to allow questions in this session. But every presenter has said that they'd be very willing to answer questions during the lunch break, and we encourage you to visit each of their posters after the session. So moving swiftly on, it's my great pleasure to introduce Dr. Kevin Sloan, who is the VP of Development Programs and Program Lead for the ALN-HTT02 program at Alnylam Pharmaceuticals. Thanks.
Thanks, Tim, and thanks everyone for the opportunity to share our work. Disclosures, I work at Alnylam, and this program is something we're doing in collaboration with Rgenta Pharmaceuticals. So we all know that Huntington's is driven by mutant Huntington. There's toxic, broadly disruptive gain of function, CG repeat expansion, and somatic instability. There's hallmark protein aggregation, widespread disruption of biology, and neurodegeneration. Now, a lot of emerging data is suggesting that both full-length mutant Huntington and the shorter exon 1 splice isoforms likely contribute to disease pathology. And Huntington-lowering approaches may offer the potential to alter the course of disease progression, but the safety and the extent of achievable clinical benefit have yet to be defined. RNA interference may be new to some of you, but it's actually quite old. It was first observed in plants in 1990 and then characterized in animals through the next 10 years or so. Alnylam was founded in 2002 to try to bring forward a new class of medicines using this approach. And we spent more than 20 years now iterating and optimizing RNA molecules to build in drug-like properties: potency, selectivity, safety, durability. And this has given rise to over 15 clinical development programs. We have 5 marketed products using this, approved drugs. And then more recently, we've turned our attention to CNS delivery to go after some of the challenging genetic targets in neurology. Now, RNA interference is a natural biological process that exists in all of us. We can engineer a synthetic, gene-specific small RNA molecule. That's our drug. And we can introduce it into the cell. Once it's in the cell, it loads into an endogenous silencing complex called RISC. And that RISC complex, we're directing it where to go. It seeks out a specific messenger RNA for destruction, which leads to reduction of protein expression downstream. Importantly, a little bit of drug goes a long way. So when we introduce that molecule, it can load and reload into the RISC complex, leading to a long duration of action. And to date, RNAi-based medicines have demonstrated potent and durable efficacy in clinic, supporting infrequent dosing regimens with acceptable safety profiles. Importantly, siRNAs are a unique class of genetic medicine that are quite distinct from antisense oligonucleotides, different chemistry, different molecules, different mechanisms inside the cells. Recently, we achieved the first human translation of RNAi in the CNS. We did this with a chemical conjugate called C16 attached to the siRNA molecules to facilitate delivery. And we did this with a molecule targeting amyloid precursor protein, or APP, which has therapeutic applications potentially in Alzheimer's disease, as well as other indications. The graph on the right over here shows clinical data from a phase 1 single ascending dose study with this platform. Here we're measuring APP protein in the CSF after a single intrathecal dose and time in the x-axis. So what you can see is after a single dose, we have rapid lowering of APP protein. That lasts for 6 months and then recovers over the next 6 months. So that's 12 months after a single dose in patients. The safety profile in this program has been very favorable so far, with no CSF abnormalities. So we've taken that same delivery platform that's unlocked our work in neurology, and we've applied it to a number of new projects. For the past number of years, we've been working on Huntington's and trying to come up with a way, really learning from all of you in the field and programs that have come before us, to find an optimal way to target Huntington's. And so we have deliberately engineered a molecule that targets a conserved sequence that lies within the first exon of the Huntington's gene. And this is important because if you think about a typical Huntington's patient, there's a wild type and a mutant allele. And by targeting this first exon, we will silence the entire full-length mutant Huntington, including the first exon, the expanded CHA repeat tract, and the downstream gene. But we will also silence any shorter fragments that contain just the first exon and the expanded CHA repeat. And by reducing all forms of mutant Huntington containing expanded polyglue tracts, we think this molecule could have the potential to limit the toxic gain of function activities and alter the course of disease progression. Now, it's important to know that because we're targeting that conserved sequence in exon 1, it's also present in the wild-type form, so we'll also engage the wild-type allele. Because of the controversy in the field about the safety of wild-type Huntington lowering, we've really taken our time and done extensive work to characterize this molecule in preclinical studies. I only have time to show you a few examples, but first is a single-dose study in primates, in monkeys. Here we're looking at protein expression in the y-axis, and then 3 months after a single dose on the left, 6 months after a single dose on the right. The light blue bars are a low dose of test drug. The dark blue bars are a medium dose. And then these are in pairs, so we're looking in cortex, hippocampus, caudate, and putamen, showing widespread distribution of silencing across these CNS regions. And if we compare the 3-month data to the 6-month data, we see durable lowering after that single dose. We do start to see recovery, which is great, but we don't want a forever drug. And the safety profile through the 6 months was very encouraging. There were no in-life neurological abnormalities, no elevations in CSF NFL, and no elevations in CSF total protein. This is now a second NHP study where we're trying to push the exposure and ask what happens when we really push Huntington lowering farther than we think we probably should. This is a multiple dose study, so 2 doses over 6 months. Huntington protein again on the y-axis, and then we're looking in cortex tissue, ground up, CSF control, low, medium, and high doses. In some animals, we're seeing greater than 90% lowering of wild-type Huntington. Again, we saw a very encouraging safety profile through 6 months with no in-life neurological abnormalities, no adverse CSF parameter changes, and we did a very extensive histopathology workup and saw absolutely no changes at the microscopic level. So this molecule has now been evaluated in 4 independent NHP data studies. Across all of them, we've seen no adverse findings, even after very deep Huntington lowering. And I'll leave it there, but if you're interested in this topic, please go visit our poster. My colleague Will Cantley is presenting, I think it's I-003, about really the totality of our learnings from these NHP studies, because it's a little different from what some of the dogma has looked like. So that profile of that molecule has been very encouraging to us. So today I'm pleased to announce that we are initiating a phase 1 study of ALN-HTT02 in adult patients with Huntington's. This is a placebo-controlled single ascending dose study to look at safety, tolerability, and PK/PD. The study population is age 25 to 70 with more than 39 CAG repeats. We're enrolling HEISS stage 2 or early stage 3. And importantly, anyone who enters the study and receives placebo in the first part is eligible to receive a dose of active study drug in the second part. So everyone who joins the study will have access to study drug. The study is initiating in the UK and Canada with additional countries planned. And we tried to partner with the community. So the protocol has been reviewed and accepted by the Enroll HD Clinical Trials Committee. It's also been reviewed and endorsed by the EHDN Executive Committee. So to summarize, our siRNA platform may offer a new approach for huntingtin lowering in the CNS. The delivery platform has already been validated in clinic through our APP program, so it's not brand new. ALN-HTT02 is an investigational RNAi therapeutic designed to durably lower all forms of mutant huntingtin, including the exon 1 fragments. And our preclinical studies suggest the potential for infrequent dosing. Based on our data in APP and the preclinical data here, we believe 6-month or less frequent dosing in clinic is what we'll do. We've shown that wild-type huntingtin lowering in the CNS appears to be well tolerated in NHPs after intrathecal dosing with this molecule. And a phase 1 study is initiating right now and will enroll later this year. Finally, we have a lot of ongoing research efforts at Alnylam, and we're exploring potential ways to apply this platform to other neurology challenges, including other aspects of Huntington's. We welcome collaborations. If anyone's interested in working with us, please reach out to us here at the meeting or contact us afterwards. With that, thank you for your attention, and thank you for all your collaboration.
Thank you very much, and it's now a great pleasure to present Dr. Serena Hung, who is Chief Medical Officer in Atalanta Therapeutics.
Good morning, everybody. I'd like to thank the organizers for allowing me the opportunity to present to you our work at Atalanta. So just a little bit of introduction about Atalanta. We're a biotech company located in Boston, Massachusetts, and we're dedicated to working on diseases in the central nervous system. We use a unique di-siRNA oligonucleotide platform to work on diseases that are previously difficult to treat in the CNS. Our current focus include Huntington's disease. We're also working on refractory severe pain, genetic developmental epileptic encephalopathy as well. So thanks to Kevin, I can save some time because he did a really good job talking about siRNAs, and we use the same pathway. But just to recap very briefly, this is a RNA interference is a naturally occurring pathway to knock down genes and protein expression. And so we're utilizing the same mechanism, basically, using a siRNA, and then it gets incorporated into the RISC, and then it go and seek out the target mRNA sequences and initiate the degradation. And historically, as I think in this room we're all aware that there are certain challenges with some oligonucleotides, namely the durability and some penetration into the deeper brain structures. And I'll show you some data to show that we are able to achieve and potentially overcome these challenges. So this is basically a graphic of what our molecule is looking like. It's different in the sense that we are putting two siRNAs together. And in the blue, you can see that it's linked by a linker. And this chemistry came from the University of Massachusetts Medical School. And it's been demonstrated that it has excellent CNS distribution, including to the spinal cord and also the deep brain structures. There's increased potency, increased durability, and improved tolerability. We're also using a technique called ionic conditioning formulation to expand the therapeutic index and overcoming some of the acute effects of some other oligonucleotides. We don't need to use conjugates or lipid nanoparticles for the delivery. So I don't think I need to go over this in any detail, but I think there is a strong rationale for targeting HTT with di-siRNA. One of the main things is that HD is a whole brain disease, and it initiates in the deep brain regions. So we want to get our drug into the deep brain regions to knock down HTT in the deep brain region to remove or reduce the driver of the disease. And I'll show you data from our NHP. So we have done some in vitro experiments and other in vivo experiments, but just in the interest of time, I'm showing you a monkey experiment. So here, I'm showing you the study design. So these monkeys get 3 different dosing regimens. One is PBS, which is the control. And then a group of monkeys get 1 single dose of 50 milligrams of ATL-101. And then their takedown is at different time points so that we can actually see the effect over the course of 6 months. And then we also want to study the effect of repeat dose of 3 doses of 50 milligrams of ATL-101. And each dose is spaced 3 months apart, and the takedown is 1 month later. And we can also look at not only durability, but we can also look at tolerability with repeat dosing. So just to show you some data from the cortex first. So I want to focus on the blue bars. And you can see in the cortex, overall, you can see a very significant lowering of Huntington. And the maximum lowering after a single dose is at 4 months. And we can achieve about 80% of Huntington lowering. And you can see the effect started coming back up at around 6 months. But even at 6 months, the lowering is still at 50%. And the purple bar are the animals with the repeat dosing. And you can see also a very dramatic lowering of Huntington. So in the caudate and the putamen, we are actually very happy to see that there is very significant lowering of Huntington as well. And the time course is very similar. So at 4 months, we see 75% lowering. And the effect started coming back up at 6 months, but we still see about 50% lowering at the 6-month time point after a single dose. And similar things, or similar amount of lowering was seen with the repeat dose. We looked at the NFL levels in the CSF over the course of the 6 months using the high-dose monkey. And we look at serial CSF collection via the intrathecal catheter. And as you can see, there's no elevation of NFL. I haven't shown them here, but we've also done some extra work using RNA-seq to look at inflammatory signatures, and we don't see anything either. So overall, in summary, NHP studies support the development of ATL-101 as a disease-modifying therapy for Huntington's disease. A single dose, 50 milligrams of di-siRNA delivered intrathecally in non-human primate produces 80% Huntington knockdown in the cortex and about 75% in the striatum. And it's maintained at 50% level at 6 months in all brain regions. There was no safety signals observed, and NFL did not increase with HTT knockdown. And in the interest of time, I also didn't show the data here, but we started doing some modeling to sort of assess what the potential effect in human would be. And in our modeling, the clinical dose of 50 milligrams can result in 6 months knockdown in the striatum with about 50% knockdown. So we plan to initiate clinical trials in HD patients in 2025. And I look forward to working with all of you in the near future. Thank you.
It's a shame they're targeting total Huntington.
Thank you very much, Serena. It's good to see you back. So moving on, next we have Professor Pai Tom, who is Professor of Neurology at the Sen Yat-sen University in China, as consultant to the exoRNA bio company. Thank you.
Thank you, Chair. Thank you, the organizer. It's my great pleasure to introduce safely assembly exome-based therapy, ER2001. ER2001 is intravenous injection siRNA for Huntington's treatment. I'm a full-time employee at Sen Yat-sen University. and consultant exoRNA. So data presented here is from preliminary analysis. So exoRNA is a clinical stage biotech based in Shanghai, China. The first product, EHDN2024, already showed promising results in human study. Recently, we got FDA and the Chinese NMPA approval. So the plan for actually is in vivo's safe assembly siRNA delivery strategy. This strategy actually includes 2 steps. The first step is design the constructs. For the construct design, we include lab 2A. That's the protein can directly bind to exosome. So it also is the basis of safe assembly. So we also have a backbone for HTT siRNA production. We also include RVG, which is a peptide for the brain targeting. So once all the construct ready, and then we inject into the body intravenously, and then the liver cell will uptake and pack the construct into the endogenous exosome and finally release into the bloodstream. Under the guidance RVG, the siRNA will pass through the blood-brain barrier, target the HTT in the brain. So we do have some animal study shows EHDN2024 very effectively lower the mutant huntingtin in both cortex and the striatum. So we also have very good excellent profiling from animal study. We can see in terms of toxicology, safety pharmacology, and the gene toxicology. So we do also have another first-in-human study. For the first human study, the army actually test the safety and found the recommended dose. We look at 10 patients. All patients receive A injection, including loading, 6 weekly. The last one is on the week 13. So the dose start from 0.04 to 0.32 milligram per kilogram. So the human data also show very good safety. We observe total 62 treatment-related AE. So AE are mild, less than grade 2. So the AE most commonly seen in the heart and in the heart, and also the lipid metabolism and the enzyme activity, liver. So in terms of PK, we can see both DNA and siRNA reach peak within an hour and return to baseline within 24 hours. So we do have some biomarker data. We see some variation in CSF NfL. So interestingly, we see EHDN2024 treatment can consistently lower by the internuclear 6. Then we do see some better clinical outcome, especially TMS. The TMS improvement is partially dose-dependent. So we do see some benefits on the cognition, especially in the tube interference. So currently we are planning for phase 1 trial both in China and the US. For China, we think we can start later this year. For US, probably we also really depends the availability of the funding or the partner. So the trial actually, the ongoing trial actually is also focused on the safety or tolerability. And then we pretty much will recruit the patient for age 25 to 50, and the early stage, if TFC score between 9 and 13. So the trial will include the phase 1A, that's the open single arm dose escalation, or phase 1B, that's the randomized blind placebo-controlled dose expansion. So all the patients will receive 6 injections and then follows up 6 months. So lastly, but not least, we really need to start a trial in Europe. So that's why I come all the way from China to attending this wonderful meeting. So I hope we can find a partner here, help us run the clinical trial in Europe. Thank you very much.
Thank you very much for coming all the way. And now we move forward to Dr. Gerard Griffioen from his Chief Scientific Officer in reMYND. Please.
Thank you. Thank you for the invitation to present here. Would like to present some of our latest results and our plans for clinical trial with a compound, a small molecule. So unlike the previous speaker, it's a small molecule that counter mutant huntingtin neurotoxicity in animal models and hopefully also in patients. However, here just a slide. This is actually not Huntington's disease, but it's a slide about Alzheimer's disease. It illustrates the ambition that we have with Huntington's disease. So we are here not only for a compound that slow down progression of disease, but we also are in for improvement of fast symptomatic benefit. And that's actually illustrated by the latest result of our AD program, where we're using a septin modulator. All about septin modulators here in this science paper. But what we see here in the animal models, that it in an LTP experiment, that we can improve the defective LTP response after 7 days of treatment in a model of Alzheimer's disease. So, oops, sorry. So that's actually shown here. We go from the blue to the green trace. And actually, although this is already very, let's say, promising, when we then recently have completed a phase 2 study in patients, and there we see actually that also here we have a symptomatic benefit. We improve memory in a cognitive test. So these non-clinical data, this non-clinical efficacy translates very well to patients. And that's what we hope to achieve also for Huntington's disease, and to develop a compound that, apart from delaying disease progression, also has symptomatic benefit. So what about Huntington's disease? What's the concept? So we have here identified a target. Unfortunately, cannot disclose the target, the identity of the target, but it's an enzyme that produces oxidative stress, reactive oxygen species. It is activated by mutant huntingtin in several ways. For instance, by providing in a striatal-specific context the substrate of this enzyme. It has an impact on expression, and so on. And what is also apparent is that reactive oxygen species do have the potential of somatic CIG expansion of mutant Huntingtons, or mutant Huntingtons. So there's actually a vicious cycle that, in the end of the day, results in exotoxicity, neurodegeneration, and symptoms. So in case you doubt the validity of this target, we did the following experiment. We used medium spiny neurons, iPSC derived from patients and from healthy controls. And the left graph shows a control CHG33 expansion tract. And here we quantify mitochondrial calcium. So mitochondrial calcium is a very important, let's say, element of excitotoxicity. And when we challenge these neurons with glutamate, we see a normal mitochondrial buffer capacity in the control cells. But very unlike is this in neurons from a juvenile patient. There, upon the addition of glutamate, so stimulus of excitotoxicity, we see really a massive activation of an accumulation of mitochondrial calcium. And this is really, I mean, very bad news, and actually would turn mitochondria into suicide machines if this is not resolved. And this is very unlike when we silence the target, then this increased accumulation, this massive accumulation, is fully rescued. So it's really a very strong mediator of the excitotoxicity driven by mutant Huntington. When we then, having this in mind, then of course the solution to this problem is let's say straightforward. We need to develop, and that's what we have done of course, a small molecule inhibitor against the target in order to prevent the onset of this toxic and noxious disease cascade. And this compound called REM004949 is a small molecule. It is currently in IND-enabling studies, and we plan to do clinical development in the course of next year. So if we then use this compound as a pharmacological tool to inhibit the target, then basically you see the same as in the silencing experiment. Also here, you see that upon pharmacological inhibition of the target, this accumulation of calcium is very strongly mitigated, very similar to what we have seen when we silence the target by genetic means. So here the efficacy is very, let's say, compelling. Then we turn to animal models. We used the ZQ175 mice. We quantified cortical striatal transmission, and as you know, in this model, the transmission is down compared to wild type. That's the gray bar. But then again, if we use this compound, treat the mice for 7 days or 28 days, doesn't matter. We have a full rescue of the cortical striatal transmission, very much as what we have seen, in fact, but then for another, of course, network in the case of Alzheimer's disease. When we do dose response studies with this, using this assay, we see that we have a dose-dependent and also exposure-dependent effect with an ED50 in mice of 0.1 milligram per kilogram, which would translate into a human equivalent dose of less than 1 milligram a day. So that is very, let's say, promising, I would say. Then we did a functional test. So these mice develop at about 12 months of age. They have a motoric deficit, as you would expect. So here, so we have used the POL test to quantify that. And you see that here, that the mutant mice treated with a vehicle have this impaired POL test performance. But then again, a month treatment with a compound fully restores the performance back to wild type levels. So also at a functional level, there's really a very promising therapeutic benefit. Biomarkers, we also looked at a couple of different biomarkers, also to be used in clinical development. TNF-alpha is a prominent one. It's a marker of inflammation. Phospho-tau, GFAP, all markers of neurodegeneration to be followed up potentially in the clinic. This is the setup of the clinical study. As I mentioned, it's still up for discussion, I must say, but this could, this is what we are thinking of. It's a phase 1 study that includes a patient cohort. We anticipate, like for the Alzheimer program, a proof of concept study where we could go for 28 days treatment in symptomatic patients, because again, also here, we expect a symptomatic benefit. We still are discussing whether we will do 28 days or longer. And of course, all the readouts that you would expect regarding pathophysiology, symptomatic function, and clinical functional readouts, such as the Q-motor score and so on. And with that, I would like to stop, and thank you.
Thank you very much, Gerard. If you have any questions for Gerard, he's available at lunchtime, and please visit his poster at I017. So we have a slight change of direction now with, I'd like to introduce Dr. Donna Finch, who is the Head of Translational and Clinical Science at Alchemab Therapeutics.
Fantastic, thanks very much, and thanks very much for the invitation to speak to you all today. As an immunologist, I've been incredibly well welcomed into your HD community, and thank you very much. Alchemab is inspired by resilience in disease, and obviously we want to acknowledge that patients living with symptoms, individuals with diagnoses, individuals at risk of disease, all show remarkable resilience every day. But I think it's worth kind of putting into context how we at Alchemab think about resilience in disease, and how we're inspired by some extraordinary examples of patients. So in cancer, it's really quite simple. These are individuals who are long-term survivors with disease. We also look at individuals who've lived to a very long age, more than 100 years, without any chronic diseases. Probably more relevant to this audience are patients who have susceptibility to neurodegenerative diseases, but who do remarkably well. So these are individuals who are at risk and perhaps have a much slower progression of disease. Now, there's obviously multiple reasons, and these include, and have been discussed at this meeting at length, the genetic modifiers, epigenetic modifiers, social, environmental. But what we're interested in, obviously, as immunologists, is there also an impact from the immune system? So could people's ability to produce antibodies impact their resilience in disease? So we all know that there's a vast capacity for each and every one of us to produce a diverse antibody response to pathogens. So everyone's response is different, and yet we can see some commonalities between people's responses when they're reacting to specific targets. We also know that in some instances, autoantibodies can also be produced. And I think most people are aware of autoimmune diseases where these autoantibodies can be pathogenic. So, for example, rheumatoid arthritis, SLE, and obviously some of the neurological diseases like anti-NMDA. But they can also be protective, and I think we're all starting to understand as immunologists that these naturally occurring autoantibodies can also be beneficial in disease. So an example here is just given in cancer, where naturally occurring anti-HER2 antibodies can be protective against the reappearance of breast cancer. So our process, which basically starts by identifying disease-resilient individuals and really being very careful about how we identify what is a really beneficial disease progression. What we do is we take all the B cells from these individuals. These are the cells that are producing antibodies, and we deep sequence. So our technology has only been enabled in the last, say, 5 years or so by advances in deep sequencing and also machine learning and the analysis of these huge data sets and the huge diversity that you see within individuals' B-cell repertoires. So what we do is we look at the sequences, we look at the sequences that could be binding to the same antigens, and we identify those that are only occurring in the resilient individuals and that are not occurring in individuals who are progressing with disease and also healthy individuals. And this is important to remove antibodies that are to common vaccinations, common pathogens, and what we're left with are a set of antibodies that could be driving resilience within the disease. We next have to discover what they're binding to, and I'm not going to be able to go through that today. That's also quite an involved process, involving lots of very sophisticated processes. But we are then able to look at what these antibodies are binding to and rationalize how they could be conferring this resilience. So in Huntington's disease, we have defined resilience. We've used the PREDICT-HD participant data, and we've looked at individuals who have CAG lengths of more than 40. These individuals also have, they have a CAP score of more than 400, and they have been showing no motor symptoms. So they are defined as having a much later than expected onset of motor symptoms within the PREDICT-HD cohort. And some of these individuals were 15, 20 years out from where they may have been expected to have their onset of disease. So again, skipping to the chase, we are still analyzing these data and looking at additional potential targets to go forward. But the first antibody that we have identified is an antibody that binds to Huntington. So this antibody is an interesting antibody. I'm not going to go through this slide because obviously this audience understands, but we are binding to multiple different forms of Huntington. We bind to the exon 1 form, we bind to fragments of Huntington. The antibody also binds, very importantly, to large aggregates and large molecular weight forms of Huntington. So we've also then gone on to do a fairly standard analysis of what this antibody is capable of. It can increase the phagocytosis of Huntington by microglia. It binds, as I said, multiple different forms of Huntington, and we've shown that it also reduces aggregation of Huntington in a cell-free seeding assay with the help of Eric Vanker. And we have also most crucially demonstrated that it reduces aggregates of Huntington observed in the R61 mouse model. And this, we think, is quite a remarkable finding. What we're currently doing is addressing this in PET ligand studies with the help of the CHDI and their PET ligand CHDI 650 to see if we can understand this further. We're repeating this in the ZQ175 model, but we also think we are going to be moving really very quickly through this stage. We're starting our non-clinical safety studies. Developability for the antibody looks fantastic, and we're moving through cell line development. And we're speaking to the networks, and I know some of you have been kind enough to participate in our KOL meetings, and we're starting to design clinical studies to see, to start to move as quickly as we can into Huntington patients. And with that, we look forward to having discussions with all of you. And we started with the patients, and I want to end by thanking very much the participants of the PREDICT-HD and those of you who've already participated in discussions with us. Thank you.
Thank you very much, and thank you to all the interesting presentation we have heard until now. And it's now a great pleasure to present the last speaker of this session, Dr. Travis Vajor, who is co-founder and Chief Scientific Officer in Rgenta Therapeutics.
Thank you. I do want to, start off by thanking Tim and Lena for the invitation to come and talk with you here today about some of the work that we're doing in Rgenta. I also, have been wandering around the neuroscience community for a long time now, and I will say that this ecosystem that has been built over the last few decades is really amazing. A lot of great people have contributed to that, and I'm glad, that I've been kind of welcomed in by the elders. So it's a great feeling, and so I probably used up a minute of my time there, and I know that, our keynote is going to tell us some really cool stuff, so I'm going to dive right in. So, as I mentioned, Rgenta is a small molecule, a drug discovery company. We're in the Boston area. We really are focused on small molecules targeting RNA, and really in historically undruggable targets. So Huntington disease is one of those that's really interesting to us. The genetics behind some of the targets is really amazing, and that has been coming out over the last few years, that repeat expansions and modifiers of those are really critical targets and very interesting targets to go after. Specifically, for us, PMS1 and MSH3 are really interesting targets to go after. Nice biology has been done at the genetics level. There's great human biology behind it as well. It is the most beautiful set of targets that I've seen in my 20-plus years in neuroscience. And then obviously there's really importance, from the Huntington, participants, the families, that there's clear unmet medical need there as well, both in the US and really all over the world, EU. So, the genetics I talked a little bit about are really strong for PMS1. So this is some human data that has been out there in addition to the modifier, the GWAS data. For PMS1, there are basically, some variants that are associated with an increase in PMS1 levels. That results in earlier onset of Huntington. On the other end of the spectrum is some really nice analysis that McAllister and group have done, where they've looked at a very large cohort. And in that cohort, those that have a loss of function of PMS1 or basically non-synonymous or damaged genes have a delayed onset and less severe symptoms. Again, those bookends of what a great target to go after. So we went into our database of potential druggable targets and said, is PMS1 and MSH3, are they druggable with our platform? Our platform is small molecules targeting RNA binding proteins. In that database, there are roughly 12,000 different target sites we can go after across 6,000 genes. In there, when we do a druggability analysis, there are roughly 1,200 of those target sites that are druggable. PMS1 has 4. So it really makes it, for us and our platform, just a brilliant target to go after. Great biology, it fits into our platform, clear unmet medical need. And so over the last few years, we've really been developing our target engine, our chemical database, as well as a lot of lead optimization, and have spent significant time in developing this. One of the cool things that we've kind of talked about a little bit already, I like this slide because it's very simple. When you think about processing of RNA, you can think about exon A stitched with exon B to make your mRNA, which goes on to, in this case, pathological protein, PMS1. We have the ability to identify these target sites where we can effectively have a small molecule, shown here in yellow, that binds to the RBP, a segment of RNA, which we can effectively insert into that pre-mRNA to yield mRNA, which itself undergoes degradation. It's destroyed. If there's no RNA, there's no protein. So it's a brilliant way to prevent that pathological protein from ever being generated. In here, a lot of work done by the MedChem group in biology. I'm just going to give a couple of little quick slides on the overall profile. This is a Huntington model of repeats where we effectively dose our compound for 30 days, and we look at how well does knockdown of RNA look. It is dose responsive in this manner. That's the left panel. And how does that track with protein? No RNA, no protein. And then when you look at these repeat and you look at essentially how do we do in these fragment analysis, you have basically the parental line, you have the vehicle where they continue to grow substantially. And then when we dose our compound, we see a nice dose response. 100% stalling, and even at 50% PMS1 knockdown, we see a 70% reduction of repeats. So again, a really nice relationship between RNA, protein, and the biology that we want to see stalling of repeats. So one of the questions that always comes up is, are we getting to our target tissue? Small molecules are beautiful from this aspect. Again, I've spent my entire career in neuroscience and small molecule drug discovery, so it's no doubt, no doubt that we can do it. I'm showing here some monkey experiments that we've done, effectively looking at different tissues. So it's really important, when we think about Huntington disease, while it is characterized primarily as a neuroscience target, there are also peripheral components, such as the heart. And this has been demonstrated and shown by various labs. Ricardo's shown some really nice data on that. So blue bar exposure, we get into the heart quite nicely, as well as different tissues, including deep brain regions such as the striatal. When we look at a chronic dosing over 28 days, we see incredible knockdown of protein. And then when we look in brains of monkeys, we see the same thing in terms of knockdown in striatal tissue, which collaborates quite nicely with the overall effects that we'd expect to see. Our clinical plan is really devised by a lot of the work that has culminated from research that people have done. I'm not going to go through this slide in too much detail because I have like 20 seconds left. But we'll do a very standard SAD, MAD, and then we'll really leverage a lot of the knowledge that has been created over the past decade, where we look at all these markers, including motor, as well as digital tools that are becoming available, and MySeq stuff, right? So really nice stuff. So effectively, I'm done. We are going to turn it over to the boss of this section and get some really good updates on future. Thank you.
So thank you very much, Travis. Some very exciting stuff there. So Travis's poster is J002, and he would be very happy to take questions during the lunch break. So very big thank you to all of our speakers for giving us a taste of what is to come. I think there's some great excitement and great potential there. So apologies once again for not giving you the opportunity to ask questions, but please approach the relevant speakers in the lunch break. So for now, please remain seated. Hold on to your hats for our keynote lecture, which is Huntington Disease Now, Mechanisms and Therapies. Alexander and Sarah, over to you.
If I have a microphone that works, I would be happy to share. Yes. Okay. Hello, everybody. Sarah, come with me, because I would like to introduce